Hello biology students, and welcome to video 3 of our DNA unit, and today's video is going to be all about DNA replication. So remember back from last video, we said that Watson and Crick were the two scientists who actually reported the structure of DNA, and they said that it was an anti-parallel double helix. And it was kind of funny, at the very end of their paper that they published, reporting the structure of DNA, they had this almost offhand statement, very casual, that said, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So in layman's terms, what they were saying is that, you know what? The fact that A always pairs with T and C always pairs with G is going to make it really, really easy for DNA to replicate or copy itself. So let's look at this a little bit more. So we know that DNA needs to be replicated before cell division happens. And remember that this happens in the S phase of the cell cycle. And the fact that strands are complementary, the fact that one strand pairs nicely with the other strands, makes it really easy for each individual strand to act as a template. In other words, so here's my DNA molecule here, and let me zoom in a little bit so we can get a little closer. So if on this side of the DNA molecule I have a T, and here I have an A, maybe C, C, G. If I'm going to build a new DNA molecule, if I'm going to replicate it and make a copy, if I already have a T on one side, so maybe I have a T right here, when I go to build my new DNA strand, I know that an A has to go there. And if I have a C already, I know a G would have to go there. So I can build the other half of the DNA based on what I already have. Okay? So this complementary structure of DNA allows for it to act as a template to copy itself. However, before this can happen, looking at this picture, what are some things that have to happen before this can happen? Well, if you look, our DNA here is unwound. So I have to first separate my DNA before this can happen. So if we're going to look at the 3 steps of replication, the first thing that has to happen is I have to separate my strands. So you can see here I have a DNA molecule, and then I split. I have one strand on one side and one on the other. Then I'm going to use the complementary base pattern that we have to add in the new nucleotides. So here I'm building the other half of one strand and building the other half of my second strand. So then at the end, I have 2 complete identical DNA molecules that I can wind back up into the double helix shape. So here's another picture showing the same thing. So first I unwind my DNA, I split it up. I use the complementary base pairing rules to synthesize a new strand of DNA. And then at the end, my new strand, my red strand that I just built, winds up with my old parent strand. And if I did this for the whole molecule, I would effectively have 2 complete copies of DNA. Okay, so there are a lot of enzymes involved in this process. We're only going to talk about a couple of them. So helicase, remember that ACE, ASC indicates it's an enzyme. And its job is to separate the 2 strands. So it breaks those hydrogen bonds between the nitrogenous bases on the 2 strands of DNA. DNA polymerase, remember polymerase, that kind of reminds me of polymer. So this is the enzyme that's going to build a DNA polymer. It's going to add the new nucleotides to the growing strand. And then finally, the last enzyme is DNA ligase. And this is kind of like glue. Every place there's a little gap or a hole, it's going to join those gaps in the backbone. So DNA is a nice solid molecule. And we'll get to each of these a little bit more as we keep going. Okay, so let's break down these steps. So the first step is strand separation. So originally my DNA molecule, one strand is bonded to the other side. But what's going to happen is the enzyme helicase, which is represented by this purple triangle right here, moves along kind of like a zipper and just unzips the DNA strand. So now I have the 2 strands are separated. And it does this by breaking the hydrogen bonds between the nitrogenous bases of the two strands and forming what we call a replication fork. So if you look, this structure over here kind of looks like a two-pronged fork, so we call that the replication fork. The spot on the DNA where this starts is called the origin of replication. So in the DNA, there's a specific sequence that helicase looks for, and when it finds this DNA sequence, it knows, aha, that's where I'm supposed to start replicating. So that's the origin of replication. So the next step is we're going to add new nucleotides. NTD stands for nucleotides, okay? And remember, it's the DNA polymerase that's going to add new nucleotides. So here, this orange, that's supposed to represent the new nucleotides that are added by DNA polymerase. So you could see here is this purple oval, that's the enzyme, and it's reading the green strand and adding the appropriate complementary nucleotide. So remember that nucleotides look very, very similar to ATP. And so the energy for this process is supplied by the energy found in the bonds of the nucleotides. It's kind of like when we break ATP, break off a phosphate group, we get some energy. If we break a phosphate group off of a nucleotide, we also get some energy. So that's where the energy is coming from. Now, this process is a little more complicated than we've talked about already, because DNA polymerase is kind of tricky, because it could actually only add a nucleotide to the 3' end of the growing strand. So in other words, it could actually only build a strand from 5' to 3'. So that's going to complicate our situation. So just keep that in mind, and we'll talk a little bit more about it in a second. Okay, so here's a kind of a closer up molecular look at DNA polymerase. So here is my old DNA strand, the strand that was green in the last picture. And what polymerase is going to do is here's the new nucleotide that it's trying to add. So it reads that the template strand is a G, and it knows it has to add the C. So it takes the nucleotide, the C bonds to the G, and then this bond right here is broken. And when I break that bond, that energy is used to attach this 5' end of my nucleotide to the 3' end of the previous nucleotide. So ultimately, I'm building my new strand from 5' to 3'. Okay. So now, this creates a huge problem, because remember, DNA is anti-parallel, which means one strand goes from 5 to 3. So if the helicase is unwinding in this direction, and my polymerase has to build from 5 to 3, this strand over here is all set. It could just read this strand and build in this direction, and as helicase unwinds, it can just continue to build. But on this other strand, we've run into a problem, because this strand goes from 3 up here to 5 down here, which means helicase is still unwinding in this direction, but my enzyme can only build a new strand. It has to start on the 5' end, and then the new strand, the 5' end, is going to be the same side as my 3' end. So it can really only build going this way. It can only read the template strand from 3 to 5. So it's building in the opposite direction than we're unwinding. Okay, so we're unwinding this way, but on this strand on the left, it can only build this way. So what ends up happening is it forms what's called Okazaki fragments. So on the right strand, the strand that we call the leading strand, the strand that we're building in the same direction as my fork is opening, we're all set. We can build in one continuous piece. But on the lagging strand, the strand over here, where we have to build this way, I'm sorry, that was a mistake. Wrong. Let me erase that. The fork is opening this way, but I have to build my new strand 5 to 3, so I have to build this way. So what ends up happening is it builds a little piece. Let me actually erase everything so you could see more clearly. I'm getting arrow happy, I'm sorry. Erase, 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 erase. Okay. So what's going to happen is I'm going to build a little piece of DNA. And then my fork is going to open up a little bit more. So we'll open up a little bit more. And then I'll build my next piece of DNA. And then it'll open up a little more. And I'll build my next piece of DNA. So I'm left with all these little short fragments that we call Okazaki fragments. But if you look, there's little gaps between the Okazaki fragments. And then finally what happens is that enzyme DNA ligase, the one that I kind of said was like glue, comes in and it fills in these gaps. So I have a nice continuous strand. So now I've created 2 DNA molecules from 1. So again, to talk a little bit more about this leading, lagging, 5', 3' stuff, remember, DNA polymerase can only build its new strand from 5 to 3. So that means it can only read the template strand 3 to 5. So up here, it could, on this top strand here, it could read 3 to 5, and it builds its new strand 5 to 3, which is in the same direction as my fork is opening. But on the bottom strand, it has to read from 3 to 5 in this direction, so it has to build in these little pieces. And then as the fork opens up more, it will build another piece and another piece, creating these Okazaki fragments. Okay, so if replication only started in one spot, which it does in some small prokaryotes, it would take forever to get through the entire DNA molecule. For example, E. coli only has 4.6 million base pairs, and it can replicate in about 1 hour. So it only starts replicating in one spot. Now, if we only started replicating in one spot on each chromosome, it would take us years to replicate, because we have base pairs in the billions. So we actually start our replication in many, many spots along the chromosome, and then they keep going in both directions until they meet each other. So we can just replicate our DNA in a few hours by doing this method. Okay. So you may have noticed earlier in the video that when I replicate my DNA, because I split it in half, each new DNA is half old, so half original, and then it's half newly synthesized. So because each new DNA has half old, half new, we call this semi-conservative. We're conserving the old DNA, but each one only has half, so we call it semi, or partially conservative. Okay, so each DNA strand has 1 template strand and 1 new daughter molecule. Then finally, the last thing we have to talk about is replication's not perfect. In fact, 1 in 10,000 times, DNA polymerase adds the wrong base. And this doesn't seem like a lot, but considering the fact that we have 6 billion bases in our genome, if we had a mistake 1 in every 10,000 times, we would have a whole lot of mutations, and that would create a lot of problems. So to prevent this, is DNA polymerase actually also has proofreading ability. It can proofread its work, and it can tell when it's made a mistake. So what it does, if, say, our template strand was something like this, okay, so this is my old DNA, and polymerase is going along, and it adds a T, and that's correct, it adds an A, and that's correct, and then it adds an A to bond with C. Well, that's wrong, but DNA polymerase will recognize that it made a mistake, it will cut out the A, and then it will go ahead and add in the correct letter. So it can proofread what it did against what it should be, remove the incorrect base. So it's kind of like catching a typo. And due to this proofreading, now we only have 1 error per every several billion, so much better rate than 1 in every 10,000. And that's it. See you in video 4.